Welcome to today's ATI seminar. The University of Alberta acknowledges that we're located on Treaty 6 territory and respects the histories, languages, and cultures of First Nations, Métis, Inuit, and all First Peoples of Canada, whose presence continues to enrich our vibrant community. We'd also like to thank Paladin for their support of the ATI seminar series. My name is Andrew Pepper, and I am absolutely thrilled and privileged to moderate today's seminar, which is centered around the impact donation has had in formulating Edmonton to become a world leader in islet transplantation, as well as a real edifice for human islet biology and diabetes research. And for over 20 years, investigators here at the University of Alberta have transformed islet transplantation into a means that is safe, reliable, and transformative therapy for patients with difficult to control type 1 diabetes. And as we reflect, this isn't the end by any means, it's not the beginning of the end, but I believe it's important to highlight the impact donation has had on the value of donation in creating such a wonderful diabetes environment here at the University of Alberta. And we need perspectives to reinforce that. So we're very fortunate today to have three amazing individuals and speakers. We're joined by Kathleen Gibson, who is an islet transplant recipient. We're also joined by Dr. James Shapiro, the leader of the clinical islet transplant program here at the University of Alberta, and Dr. Patrick McDonald, who is the director of the Human Islet Research Corps, who will speak towards how human islet donation has allowed for cutting edge human islet biology and diabetes research. So it's with my great pleasure to introduce our first speaker, Kathleen Gibson. So Kathleen Gibson has lived well with type 1 diabetes for almost 28 years, and she acknowledges with gratitude the generous gifts she has received from 3 islet cell donors over the past 10 years. And Kathleen is an advocate for living well with any chronic disease and is a registered dietitian and certified diabetes educator. And we're absolutely thrilled that you're here today and sharing your story with us.
Thank you, Andrew. So hello, everyone. My name is Kathleen Gibson, and I am a 3-time islet cell recipient. So I was diagnosed with type 1 diabetes at the age of 27 in August of 1994. It came as a shock to my knowledge. I didn't know anybody with diabetes. I didn't know what diabetes was. And it meant I had to do a lot of juggling in my life and changing things. But I was an environmental scientist, so I approached it from a scientific perspective and figured out how to make it all work. I started on multiple daily injections of NPH in Toronto. And as new insulins came, I got transitioned to new insulins. And I got an insulin pump in 2000 when I worked for the University of Pittsburgh. And I got a second pump in 2006 when I began my employment with my current employer, the Government of Canada. I had to self-fund my insulin pump during my nutrition degree, which came after an environmental science degree. And I've always had a fair amount of sensitivity to insulin, and so lows have been a frequent part of my life. In fact, in order to get my second pump, my endocrinologist had to write the term brittle diabetes, which is always hard to acknowledge when you think you're doing well, but it's still hard. In 2011, Dr. Senior came to talk to the Edmonton chapter of the Diabetes Professional Section of Diabetes Canada, and was talking about the recent progress within the islet cell program. And it was very exciting. And at that point, I thought, you know what, I'm gonna try. So I put my name on the list and I went through all the workup and I was accepted. And it was a really exciting time. The workup process to get an islet cell is quite extensive, and I learned a great deal about my own health. I learned I have an ischemic area on my heart that doesn't seem to get in the way of anything I do, but I didn't know it was there. One of the drawbacks, my endocrinologist at the time, when I put forward that I would like to pursue an islet transplant, then dropped me as a patient and said, you know what, they will take care of you. And so I entered into the islet cell program with that in mind, that they will take care of me with all things. And so I'm going to plant that seed and we'll come back to it. I've always been a supporter of research. I participated in research during my nutrition degree, looking at caffeine and type 1 diabetes. And so when the opportunity came for my first transplant to be part of a research protocol, I jumped on it, and it was using a different immunosuppression agent. And so it was delivered by injection, it was belatacept. And so that was my first journey with islet cell transplant. The transplant failed at the end of May 2012, so about 4 months. And that was really hard. You know, when things go bad, you always think, well, at least I do, as somebody living with type 1 diabetes, what have I done? How have I failed myself? I think what it was that the transplant medication wasn't able to support me on a maintenance dose. But it was hard after all of this promise and excitement of your first transplant. So the team put me on tacrolimus in June of 2012 in order to preserve whatever islet cells they possibly could preserve. And so through the summer, there was lots of conversations. And then I was listed again for my second transplant in early September of 2012. And I've been fortunate with my first 2 transplants in that I was listed, and basically 2 or 3 days later, I got the call to go and get a transplant. And so I didn't have a lot of experience of waiting between being listed and getting the call. The waiting can be hard to get on the list. And I had some experiences with some not great communication with the transplant coordinators. In fact, I got some very mixed messaging depending which transplant coordinator I spoke to. And so that element of chaos sort of got in the way a little bit, but it all worked out and I had my second transplant. Having been on tacrolimus, we know that it impacts the kidneys, and it impacted my kidneys. So I now live with stage 3B chronic kidney disease because of the tacrolimus. It doesn't get in my way of life. It's been stable for 10 years. It only sort of raises its head if I need to look at any new medications and how they might impact me. So my marriage ended in October 2012. And so I was really grateful for the transplant team and having psychologists to help me deal with new transplant, new protocol, since I was officially on the Edmonton protocol for that one, along with a significant life change. And so having that comprehensive healthcare team is really beneficial, and not being able to address my life as it was very helpful in the fall of 2012. I want to think I've lived a good life with diabetes and lived a good life with being a transplant recipient. One of the gifts that having had these transplants has given me is the opportunity to travel. I am a registered dietitian and a certified diabetes educator. I work for the Government of Canada with many Indigenous communities in Alberta. So at the time, I was providing support to nations who are a 10-hour drive straight north of Edmonton. And having that transplant gave me the confidence to do that drive by myself. Prior to transplant, I was always part of a team and went out with other people. And having that freedom to just go out and do what I needed to do in my workplace was hugely beneficial. And then the government of Alberta gave me a 5-year driver's license. And that was the first time of having a 5-year driver's license since my diagnosis. There's a freedom from the rigid eating and activity protocols that go along with managing diabetes. It's helpful in terms of being able to eat later, eat earlier. And that was a real gift in terms of being able to connect with people on different schedules. During 2014, I was able to support someone else to get an islet cell transplant. One of the program requirements is that you have a support person. And for many people, it is a spouse. And it was my spouse for my first 2 transplants. And so being able to step up and provide that support to somebody who didn't have a partner or didn't have family close by was very meaningful. And we're still in contact today, and he's really rocking his islet cells. So that's awesome. Fast forward to February of 2016, I got the flu, and it took out a lot of my islet cells to the point where I needed to take insulin again, and a fair amount of insulin. And so I was listed and got my 3rd transplant on Good Friday of 2016. So that was awesome. I was able to travel off the continent for the first time since 1985 in 2017. And I went to Europe by myself for a month, and that was hugely exciting. You can probably do the math and figure out my age. And so having now had 3 islet cell transplants, I'm going through peri and menopause and postmenopause. And for just the reality of type 1 diabetes and transplant and menopause, it's largely uncharted territory. So things changed in my body, and I had to just start taking long-acting insulin in the fall of 2018. At this point in time, recognizing the damage that tacrolimus was doing to my kidneys, I was also given sirolimus at the same time. And it impacted my skin greatly. And for 3 months, it looked like I had a sunburn over my whole body, and then it kind of calmed down. But it has gifted me with swelling in my leg. And now my life includes blood pressure medications and compression socks, which is, it is what it is. I now take multiple daily injections, Tresiba, and along with NovoRapid. My total daily dose is between 11 and 14 units, so not very much. So I have some functioning islet cells. I have some exogenous insulin. I'm on target 87% of the time, and my last A1C was 6.3. So I take all those things as really positive and doing well. I don't really have any complications from diabetes. The current complications I have are related to transplant medication. And who knows where I would be on a complication journey if I hadn't had a transplant? I don't know, but my life is far richer for having had the transplants. One of my concerns is I no longer fit into any neat box when it comes to living with type 1 diabetes. I'm not really type 1 because I have functioning islet cells. I'm kind of closer to type 2, but I'm not type 2. And so it's often challenging working with healthcare providers because I don't fit neatly anywhere. My own family doctor called me complicated, which I challenged him on, saying, you know what, my health is complicated, but I really am not. I'm quite a strong self-advocate, and I don't think that makes me complicated. And I apologize for my voice. I got the cold, magic cold that's going around came to me yesterday. So it's important to know who you are and to support yourself as a person living with type 1 diabetes and as a person who has received transplants. It's important to recognize the role of all of the healthcare providers. What I have learned, perhaps the hard way, is that islet cell coordinators, islet transplant coordinators, are not diabetes educators. They're there to support your transplant, but not really to support you with diabetes. So for the first time in 10 years, I've had to find another endocrinologist, and that's very interesting in the current landscape of healthcare in Alberta. So I work hard to maintain living well with diabetes and a transplant. I think that has been my journey all along through diabetes, and maybe because I was diagnosed at 27, I've been able to approach it with a different perspective. I offer some suggestions moving forward that if you have patients who are exploring islet cell transplant, really be clear with what the role of the transplant team is and the need to have a separate diabetes sort of care, and that be clear on the coordination of care. I would, as a healthcare provider, I would actively promote the peer support groups for people living with diabetes, transplants, various other chronic conditions. Those groups offer a source of support for people living with a chronic condition or living with a transplant. And most of the conversation that happens is around, well, you need to connect with your coordinator or you need to call your nurse. It isn't, there's no, we don't try to diagnose each other or provide medical advice. The third thing I would recommend is, if you want to know how your clinic is doing or how your team is doing, actively seek the feedback of your patients. It should not be a passive process. And my last recommendation is, my time is valuable, your time is valuable. I really hope the virtual meetings can be part of some kind of hybrid healthcare system moving forward. But I really thank you for giving me this opportunity to share my story today. I know I've gone through with the last 10 years in a blur, but I hope there's been some nuggets of information. And I'm really grateful for the decision I've made to have these transplants. And I'm not sure what my future will hold. Right now, I'm holding steady.
Thank you so much, Kathleen. We're exceedingly grateful for you to be here today and sharing your enlightening and inspirational perspectives and experience. And I very much look forward to your insights during the Q&A session. Well, our next speaker is Dr. James Shapiro. I'm pleased to introduce him. Dr. Shapiro was born in Leeds, England, a son of a family doctor. He developed a long-standing interest in liver and islet cell transplantation as a medical student. He has been on faculty of the U of A since 1998, where he holds a Canadian Research Chair in Transplantation Surgery and Regenerative Medicine. He was a lead investigator on the famous Edmonton Protocol for cell transplant treatment for diabetes. He is the first in Canada to start clinical trials with human stem cell derived insulin secreting cells, and most recently carried out the first in human CRISPR gene edited stem cell islet progenitor transplants without immunosuppression in collaboration with ViaCyte and CRISPR Therapeutics. His translational lab is focused on generating autologous iPS islets for transplantations in patients without immunosuppression, and his team and collaborators are working on scaling up and manufacturing efficiencies to make this scalable and possible. He is the director of liver transplant, living donor liver transplant, islet transplant programs at the University of Alberta. He has an H-index of 73, which is exceedingly high. His many awards include the Hunterian, a medal from the Royal College of Surgeons of England, and gold medals from the Governors General and from the Royal College of Physicians and Surgeons of Canada. He is a fellow of the Royal Society of Canada and is a fellow of the Canadian Academy of Health Sciences. So thank you, Dr. Shapiro, for being here. And we look forward to your perspective.
Those are my disclosures. History and future of clinical islet transplant program. I'm going to take you on a whirlwind tour of where we've gone and where we hope to go to. So the objective is to discuss the 20-year outcomes of islet transplantation, discuss how much runway there is left to land will take off with islet transplantation, thinking about the supply of cells and thinking about how we can minimize and eventually eliminate the need for immunosuppression. So we're just over 100 years since the discovery of insulin, as you all know. It's a wonderful Canadian discovery. We've almost 50 years since the discovery that islet transplantation can reverse diabetes in mice, a paper from Paul Lacey and his team demonstrating that when you put those cells into the portal vein, they can reverse diabetes. And I think today we have tangible proof that injecting insulin versus an islet transplantation can potentially provide better metabolic control so long as those cells survive. Insulin is always going to be limited by injection under the skin. Those bolus insulins are absorbed in such a variable fashion that it's almost impossible to stabilize glycemic control to the need that is level that was expected, even despite many advances in modern technologies. So we're looking here with islet cell and future cell transplants as a biological solution to a biological disease. We began this experiment, if you will, 22 years ago on the 11th of March, 1999. Here we are in the basement laboratory of the SMRI at 11 o'clock at night with Ray Rajot calling the digestion, Jonathan Lakey shaking the chamber. And we isolated islets for the first 7 patients, all of whom were able to come off insulin at the time we published that report. And we've moved on since. A number of different centers have replicated and advanced these protocols, such as the ones from Bernard Herring with single donor islet transplant engraftment success with some anti-inflammatory medications. The group from Jose Oberholzer, who trained with us for a while and went to Chicago and replicated the islet success with a number of different protocols using anti-inflammatory and a GLP-1 long-acting analog. And Camilo Ricordi from Miami, who really developed the Ricordi chamber and the methods for mass isolation of human islets, a method that we use in standard form today. And here's Tatsuya Kin and Doug O'Gorman and their team, incredible team, working day and night, isolating high-quality islets for clinical transplantation. And again, as Andrew pointed out, acknowledging the donor support that we've had from right across the country, not only in the HOPE program in Alberta, but the coordination of that between all programs across the country, ensuring that the pancreas organs are either sent for us for clinical transplant or now to Pat McDonald's lab for research. So the processes involved with the islet isolation and the GMP manufacture of human islets has been a big task in itself. And you can see the documentation required to get approval through the NIH at 8 different facilities for some of the trials that we carried out. The transplant side of it is fairly straightforward. It's carried out by our expert interventional radiologist, Richard Owens, back there, injecting local anesthetic in an awake patient and then simply dripping cells into the liver and now plugging the tract very effectively with an Avitine paste to prevent risk of bleeding afterwards. It's a very routine, very straightforward transplant that doesn't require surgery. We've been involved with several different consortium trials. The first was the multi-center trial of the Edmonton Protocol published in 2006 in the New England Journal of Medicine. A phase 3 trial we were involved in with the NIH and with a number of centers in the US and in Sweden. And that was a very successful trial demonstrating that we could maintain hemoglobin A1C less than 7% and protect patients from severe hypoglycemia with islet transplantation. And a very similar trial, CIT06, islet after kidney transplantation, again, demonstrating very similar findings. A group in Europe was also able to put together this Trimeco trial, a multi-center randomized control trial comparing islet transplantation with insulin, demonstrating superior glycemic control, positive C-peptide, and reduction in hemoglobin A1C only achieved with the islet transplant procedure. And a group from Edinburgh and the UK team, Shireen Forbes being the first author who we've worked very closely with here in Edmonton, demonstrating in their findings that the shorter time of transplants between the first and the subsequent transplant did lead to some improved survival in the first year afterwards, but didn't seem to alter the long-term outcomes. So what about the 20-year outcomes with this kind of approach? Well, we recently published our 20-year outcomes, and I'll emphasize This has been a massive effort with a huge team of outstanding individuals that have contributed to the success of this program, from donor and recipient coordinators, interventional radiology teams, the islet isolation staff, our colleagues in clinical endocrinology that have now become transplant surgeons in their own right, and my other surgical colleagues, an amazing team that have taken this together with our researchers to advance it. And I also acknowledge here the transplant fellows that have worked so hard in our program over the years, and you can see many of them have gone to disparate parts of the world, but some stayed with us. Khalid Dajani, Julia Imamali now is in California. Peter Senior, of course, is our transplant endocrinologist with the Islet Transplant program, a medical director, and this has been a great journey and privilege in itself in training these individuals. So Braulio Marfolgaza, who is about to defend his PhD in a couple of weeks, has done an amazing job pulling together the 20-year data in our patients. And I'll just very briefly walk you through some of that data. I don't have time to go through it all, but demonstrating really, he grouped the patients into those that had sustained graft function over time for 90% of their time versus those that lost function at various time points. And you can see in the red lines here in the paper, in the Lancet Diabetes paper, demonstrating sustained C-peptide across to 20 years with excellent beta-2 scores in that group. And if we summarize the C-peptide for all patients, this is a median for all patients across time, you can see that we're able to sustain high levels of C-peptide, or at least modest levels of C-peptide across 20 years. Using the beta-2 score that Peter Senior and Shireen Forbes developed, we can look at the beta-2 score over the course of time and see here that that does help to protect the islet function. And we can also see that the hypo score, the ability index, and the Clark score are markedly suppressed to close to zero in the early and late phases across the 20 years here, the hypo score medians showing very low rates of risk of hypoglycemia in patients receiving islet transplant. You heard from Kathleen and the challenges of the immunosuppression and also some of the potential benefits of being involved with islet transplantation, and Nina Green, patient 252, enjoying islet transplants and the period of freedom without insulin. So we're able to achieve periods of insulin independence in patients, and that is not our focus. The focus is to stabilize glycemic control, but we're able to achieve periods of insulin independence in 78% of our patients coming through, and this is a large cohort of patients now over the course of time. But if we look over the course of time for durability, we see that most patients with islet transplantation go back onto at least small amounts of insulin over time. And that's certainly very different with a pancreas transplant in our center, where 50% of patients will remain insulin-free at 20 years. So that offers an opportunity for research and advancement too. In the laboratory about 12 years ago, Mike McCall, who was doing a PhD in our lab, discovered that the use of two anti-inflammatory agents, anakinra and etanercept, improved the survival and function of a marginal mass of human islets in mice transplanted into immunodeficient mice. And the way this worked was it markedly suppressed cell death in the first 24 hours. We moved that program across to the clinic, and we found in the Lancet paper that this improved the odds ratio by 7.5 times when we used the combination of those two agents. And really quite incredible when you think about it. We're giving two anti-inflammatory drugs, and they're having impact out to 20 years later when they're only given for the first 2 weeks after transplant. So clearly we can alter the long-term course over things. Kathleen mentioned her hemoglobin A1C, and you can see the hemoglobin A1C of all our patients here, those with sustained graft function in the blue lines here, with A1Cs between 6 and 6.5 sustained over 20 years. And it's that what gives us hope that we can control potentially and potentially reverse some of the complications of diabetes. And this is the median A1Cs for all patients, all comers. If we look here at the survival of patients, we see that the patient survival is 74% at 20 years. And the patient graft survival is 50% at 20 years measured by C-peptide secretion. If we put the patient survival in context, some of our patients are in their 60s or upwards when they come into transplant. So this is the impact of diabetes as well as the potential risk long-term of chronic immunosuppression. If we compare islet transplant outcomes with patients with in different trials with diabetes, and look at the death rate per 1,000 patient years, you can see that islet transplantation compares favorably. And certainly it would appear, from at least from this kind of data, although we don't have a true control group in the Edmonton trials, that we are not doing excess harm for patients. We see that there are increased rates of suicide in some of our patients, recognizing some of the psychological stresses that these kind of patients undergo because of the severe swings in blood sugar. So here is a study demonstrating that patients with severe hypoglycemia have a 3.4-fold increased risk of death. And these are the patients that we've targeted for the islet transplant thus far to try to minimize that risk. Suicide, we've mentioned, is a big challenge for these kind of patients, and some of it is related to the diabetes, some of it is related to the care, and perhaps some of it is related to the transplant expectations and potential disappointments involved. In the US, it's not been so easy to get approval for islet transplantation. There's been these trials that we've mentioned, but the biological license in the US has been enigmatic, shall we say. A number of centers have applied and found it a very difficult and expensive process to secure a process with the FDA to get a biological license for approval. We were able to do that with Health Canada and through the government of Alberta, I think, very readily in 2001. We were very fortunate in that. One group, Jose Oberholzer, has developed a company to try to move forward with the biological license in the US, and they were successful in that so far, and we'll have to wait and see how that goes. In terms of looking for sites other than the liver to implant islets, there's been a lot of work in that area. We've been involved with some of it. We replicated Camilla Ricordi's omental islet transplants with a laparoscopic approach, placing islets with a minimal invasive procedure onto the surface of the omentum. But in our hands, we didn't have the same success that Camillo had, and barely were able to get detectable C-peptide in our patients, although we did have some. And Francisco Sordek was also trying to replicate that and had similar challenges. So more recently, Kevin Verhoff in the lab has put together this publication just about to come out in Transplantation, looking at all of our patients receiving extrahepatic islet transplants, be it subcutaneous, be it gastric submucosal, or in the other extrahepatic sites, subcutaneous. And we find that those patients have minimal to negligible C-peptide or the omental. But when we give them a subsequent intraportal islet transplant, we are able to restore C-peptide secretion in the standard route. And this suggests that while there's interest in the extrahepatic sites, we've still got a long, long way to go. So we've been working with Mingling Ma with this traffic type device, with Braulio working on that, went to Cornell to learn the techniques and has been able to advance it here. And we've also been combining that more recently with some other approaches. We're interested in this paper that Pat McDonald was a co-author on, where they're transplanting essentially islets with functional blood vessels to improve vascularity. That might be one way around improving the favorability of some of these sites. Andrew Pepper carried out this remarkable study with the prevascularization of a subcutaneous site, placing a plastic rod under the skin to improve these kind of new blood vessels, and was able to sustain a survival of islets and stem cells for very, very long periods of time in mice. And most recently, we've just taken this forward after about 3 or 4 years of planning delayed by COVID to a first-in-human clinical trial. And here we are in an awake patient deploying under local anesthetic the plastic nylon catheter under the skin, which is meant to be there for about a month or so. First patients had that now for about 6.5 weeks. And just a couple of days ago, this is the first transplant ever done, I think, on 3G2. That's Dr. Blair Anderson, myself, and Khaled Dajani, retrieving that plastic device, cannulating it with a guide wire, and then down that tract placing the islets, a proportion of the islets, I should say. So this patient received 500,000 islets, or just over, into the portal vein, and about 3,000 islets were placed in the subcutaneous space. And the idea here is that we'll take biopsies of the skin at weeks 2, 4, 8, and 12, and look under the microscope, see if we have islets surviving histologically, And similarly, at the same time, we're working with Phil Halloran's group to look at some of the gene arrays and see what's activated or not in this site. And this is going to be very interesting data as we move forward. So very simple approach, and potentially this could be scaled up. Right now we're using 8 centimeter long tiny tubes, but we could potentially put multiple tubes like that as an array, if you will, down the leg and down the arms and on the abdominal wall if we wanted to deploy the entire islet mass into that site. So we made our counter incision on the other end so that we would be able to find it at the end of the day. So finally, maybe just moving on to immunosuppression, we're trying to eliminate the side effects like Kathleen was describing with the use of sirolimus. We were definitely using that drug at too high dose originally. There's many other agents that we've been exploring recently, the most recent of which is this AT1501, which is a CD40 ligand antibody, for which we have clinical approval to move forward, but no patients, in fact, to enroll at the present time. But we always remember when we're dealing with this that we have to control both rejection and autoimmunity as we play around with the regimens. So Bob here is exchanging insulin happily for a few pills every day. We know that those pills, as Kathleen so well described, cancer, infections, and renal failure can be side effects, particularly with high-dose tacrolimus. So we're exploring a number of other approaches with this living immunosuppression type approach with Treg therapies, working with Chrissy Tang and Jeff Bluestone in UCSF, reinfusing autologous polyclonally expanded Tregs into our patients. And we've moved forward so far and able to at least detect some survival of these by deuterium labeling in the patients so far, but that's an ongoing trial. So the question is, how much runway is left for islet transplantation? We've proved that the islet transplant can be beneficial, but it's certainly not a cure at the present time, but could be if we eliminated the constraint of the donor source and we eliminated the need for immunosuppression. So how much runway is left? Is there room to land? Is there room for taking off with islet transplantation when there's so much excitement now with stem cells and other approaches? So how can we fix the islet supply? Xenotransplantation has been very much on the horizon, but it's worth remembering back 30, 40 years ago when Carl Groth carried out the very first transplants of pig islets into patients. These were fetal pig islets placed under the kidney capsule of patients undergoing a standard kidney transplant. And he was able to detect pig C-peptide in the urine of these patients out to 300 days afterwards. Now, more recently, there's been a lot of interest in CRISPR gene editing of these porcine organs of all sorts, and potentially that might be one way forward. I know Greg is very interested in that and has been potentially taking forward with his neonatal porcine islet protocols to potentially move that forward at some stage soon. In the last year, a lot of progress. So here was a patient receiving this was actually an organ donor, a brain dead organ donor, where a pig kidney was implanted on the vessels of the groin and survived for over 56 hours and made urine and didn't suffer hyperacute rejection after those gene edits. And here's another patient that had a heart transplant success.
That is the heart of a genetically engineered pig.
The organ looks perfect.
And he survived for over 3.5 weeks with that pig heart that had been gene edited before succumbing. Well, we've been interested for quite a while in the use of stem cells to replace the need for organ donors. There's been amazing progress with a number of scientists in Canada and the US, Tim Kiefer in Vancouver, Christina Nostro in Toronto, Doug Melton in Boston, Matthias Hebrock in UCSF, really making amazing progress. And the group in Viasite generating these stage 4 cells generated from human embryonic stem cell derived islets with growth factors. We were very fortunate in carrying out the very first trials in Canada about 9 years ago, planting these devices and PECO1 cells derived from embryonic stem cells into patients with type 1 diabetes, with and without immunosuppression. And most recently, last year in December, we reported our findings with about 1/3 of patients having detectable C-peptide at low levels, no teratoma and no risk seemingly to these patients that tolerated the procedure very well. The group Vertex and Doug Melton moved forward with a first-in-human clinical trial and implanted their stage 6 islet cells derived from embryonic stem cells into a patient, or now 3 patients into the portal vein here. So very impressive. Initially, they were able to achieve 91% decrease in insulin, and then that patient, Brian, was able to come off insulin entirely with full dose immunosuppression in this ongoing trial, which has currently been placed on hold by the FDA and led to a drop in the stock prices. We've been working recently with CRISPR Therapeutics and ViaCyte on a gene-edited PECO1 line that has 2 deletions and 2 additions to prevent immunoactivation and reduce inflammation in the transplant, and that's an ongoing trial. We're very fortunate here in the operating rooms at University Hospital to be able to do the first transplant of this in an ongoing trial. We have to wait and see whether those gene edits were effective. My own lab's been very interested in personalized iPS-derived cells, autologous islets made from patients' blood. This seemed like it was almost impossible initially, like alchemy. But the work of Shinya Yamanaka and his Yamanaka factors have enabled the transition of blood cells, in our case, into making islet cells with direct differentiation using small molecule growth factors, and using a very complex recipe now shown here. And Dadich and the team have been able to replicate these and advance on these protocols in the lab very successfully now. And we've been able to use these mini bioreactors to generate islets to transplant and reverse diabetes in mice. That's the team involved. These are the iPS cells that generate islets. These are dynamic time-lapse photography showing these colonies growing. And these are the products that we're able to generate, stage 6 islet-like cell products that have been purified either with acetylcholine or by disaggregation, reaggregation, to make sure we don't have any off targets. Pat McDonald's been studying our cells in great detail, and we're very grateful to him and his team for doing that, demonstrating at least there is early and late phase exocytosis and calcium fluxes, and we're able to look at dynamic glucose and insulin release from low and high glucose with these kind of cells, and reverse diabetes in mice with islets essentially made from patients' blood with patients with type 1 and type 2 diabetes. So we're very excited in taking this kind of work forward. Greg Corbett has this ultra clean room GMP facility that we might be able to replicate this work in our first patients soon. This is all possible. There's been a lot of hype and hope, I think, but at the same time, massive progress in the field. You know, if you think about your cell phone, it used to be not just a brick, but a suitcase you'd carry around, and now you have a tiny thing you can wear on your wrist. So this is the revolution. It's moving forward. It's possible. The use of maybe these kind of technologies from Celino that use laser capture and destruction to eliminate unnecessary clones that we want to get rid of, the ability to scale this up in existing technology using these kind of bioreactors, the work from tree frog therapeutics that allows these to be mass manufactured in vats, the work from Lonza that allows automation of these in an orchard where you could potentially grow human cells for regenerative medicine of all sizes and sizes. Do we generate a universal cell bank or an autologous product? I don't have time this morning to go through these, but we can discuss that in detail, of course. But I think the point is there's a spark here. There's hope on the horizon. A new day has begun in cell therapy, and we perhaps no longer need to rely on organ donors and on immunosuppression to take this forward. So I'm going to stop there, and thank you very much indeed.
Thank you, Dr. Shapiro, for first off your kind words, and also congratulations to you and your team, a true tour de force in your approaches to improve the lives of patients living with type 1 diabetes. It's my great pleasure to introduce our final speaker, Dr. Patrick McDonald. Dr. McDonald has been studying pancreatic islets for nearly 25 years. He has completed his PhD at the University of Toronto in 2003, and then a postdoctoral fellows at Lund University in Sweden and the University of Oxford. In 2006, he established his laboratory in the Department of Pharmacology in the Alberta Transplant Institute at the University of Alberta. Since that time, he has contributed to our understanding of pancreatic hormone secretion and has established a research program focused on understanding the biology of islet cells from human donors with and without diabetes. Thank you, Pat, for being here.
Great, thanks, Andrew. And I want to thank also Kathleen for that great perspective, and I really appreciated hearing your story. And thanks to James as well for the overview of the history and the future of islet transplant. I have no conflicts to declare. And what I'm going to do, and I don't think it will take all too long, is really just try to give a flavor of the work that we're doing to promote islet research and and show you what can be done with donor organs for research and how this will ultimately push translational work forward. And Dr. Shapiro, I think, mentioned this study, and I'm using this really as the what I think is the current cutting edge for transplantation in diabetes, and that is the work showing the production of insulin and C-peptide by stem cells transplanted into patients with type 1 diabetes, and of course this is where we are today. But I'm sure that most people on this call would appreciate that this is preceded by decades and decades of foundational basic science research. First on the, I guess, more general developmental biology of the pancreas, understanding where islet cells come from, the genetic programs that regulate that, and that work started as early as the 1950s. And then in the mid-1990s, the first attempts to make insulin producing cells in a dish from stem cells. The first attempts being around the time when I was in graduate school, and it was pretty dismal. And at that time, I thought that, well, this is never going to work. And of course, here we are today, decades on, showing that it is likely to be a viable approach for diabetes treatment. And so, I was putting together this little graphic and trying to think about, well, how can I sort of show where I think we fit in to this story, and how can we help push transplant and diabetes treatment and research forward. I really think that my role is to work to expand that and broaden the base of foundational knowledge that contributes to progression in translational and clinical work. And so by broadening that base, we can build a taller pyramid and go from where we are today to where we want to be tomorrow. And this applies not just to transplantation, but diabetes prevention, other treatments, and so on. Of course, we don't really know where this peak should be or where we want to get to, but we keep trying to push upwards. So we're trying to promote and facilitate research in diabetes and islets and islet biology. And I guess it was about 10 years ago now, we started to think, well, how can we help or help others to answer some of the important questions in islet biology and diabetes? How can we work to expand the overall base of knowledge that can be built upon? Of course, you know that in clinical transplantation, whether it be islet transplant or whole pancreas transplant, organs from across the country, generously donated by organ donors and their families, can be targeted to clinical use. Sometimes in islet isolation, the islets don't go for clinical use for various reasons, and they can be used for research. And I really would want to say that the clinical islet program that Dr. Shapiro described has been an extremely strong supporter of islet research over many, many years. And that's, I'd like to point out Tetsuya Kin and Doug O'Gorman, who have contributed to that. But there are many organs that don't enter the clinical program and may have research consent. And we established a program in parallel and in collaboration with the clinical group to accept those organs with research consent not used clinically into our program, which we call the Alberta Diabetes Institute Islet Core. There's a few pictures to the right there. This is James Lyon, who's our lead technician, works with us, but also in the clinical group. So we process these pancreas for islet isolation for research only. And once we do that, we distribute tissue across Alberta, Canada, and internationally. If you want to see more information about our research distribution program, you can find a recent annual report here, which describes all of our ongoings and activities. But a few things I will say about the program. I think I've mentioned these first 2 points. One of the advantages and one of the initial goals of the research program was to isolate islets from a wide range of organ donors, as wide as we can possibly get. And this includes the very young to the very old. We've isolated and studied islets from organ donors as young as, I think, 10 weeks, may have been the youngest. We study, of course, organ donors without diabetes or no diabetes, but also organ donors who have lived with type 1 or type 2 diabetes, or organ donors who may be lean or obese. There are a lot of important scientific questions that can be answered by studying tissue from these individuals. The other thing to note about a research-specific program is a focus on accessibility and affordability. Someone has to pay for all of this, of course, and if we can process organs in as economically efficient a manner as possible, we can increase affordability for downstream researchers and increase the amount of research that can be done overall. Over the past 10 years, this research-specific islet program has grown considerably. We now supply research tissue to over 140 groups and institutions around the world. You can see a map at the bottom there. We have institutional agreements, for example, with places like Stanford, where we supply the Stanford Diabetes Research Center with research tissue. And so these groups, of course, have then done all kinds of work on this tissue. We've also expanded from providing freshly isolated islets to providing bank tissue for research, blocks and slides, of course, and other tissue samples, such as intestine, spleen, pancreatic lymph nodes, and so on. So I'm not going to take up too much more time because I want to make sure we have time for questions. So just 2 quick research examples. We know, of course, in type 1 diabetes that islets and insulin-producing beta cells are destroyed through an autoimmune reaction. But we've known actually for about 15 years that destruction of insulin-producing cells in most people with type 1 diabetes is not complete. And in fact, most people with type 1 diabetes have some insulin-producing cells. This is a study here from the Joslin Institute showing that even in people who have had type 1 diabetes for as many as 50 years, still have some insulin-producing cells, either scattered individually throughout the pancreas or even some islets with strong insulin staining that looks, aside from that amyloid, looks fairly normal. We know most people with type 1 diabetes have some small amounts of insulin in circulation that may be detectable. Now, this is, of course, not sufficient to control blood sugar, but it's interesting from a scientific perspective, because if you can isolate those cells and study them, you can maybe find out what's so special about them. So this is some work that's ongoing in our lab to isolate islet cells from organ donors with type 1 diabetes. This is an islet prep, or I shouldn't say an islet prep, I should say a tissue prep from an organ donor with type 1 diabetes. And you see some insulin positive tissue fragments in that isolation. And these are just a couple of different donors. Here showing mostly glucagon cells with a couple of scattered insulin positive cells. And in this donor here, quite a few, at least in this fragment of tissue, quite a few insulin positive cells. So we're working to study the individual insulin producing and glucagon or other hormone producing cells from organ donors with type 1 diabetes and compare, for example, their activity, which we're looking at here, and their gene expression. So we want to know what's so special about these insulin producing cells that are still hanging around in type 1 diabetes. Are they protected from the immune system, or are they newly born? Did they appear after the establishment of type 1 diabetes? And then actually Dr. Shapiro touched on this. Since we're talking about transplantation here, the research program can contribute to knowledge in improving transplantation. For example, by studying the importance of vascularization in transplant outcomes. Dr. Shapiro mentioned this study here, where we worked with Manuel Empofo in Germany, and he was integrating microvessel fragments into the islet graft. But similar work has also been done, again, supported by our research program this is from Christina Nostro in Toronto, showing something very similar with either human primary islet cells or stem cell derived beta cells. And that's really just a taste of the kind of basic science that can be done and supported by a human islet research program. There are all kinds of other studies that are going on through this network that we've established across the world. And of course, a big thank you to those who work in our lab to promote this work, but also the clinical islet group. We wouldn't be doing this kind of work if it wasn't for them. And the same goes doubly so for organ donors and their families.
Thank you, Dr. McDonald, for really highlighting the importance and value human islet donation has on that fundamental research from both individuals living with diabetes and without. So hopefully the panel can stick around to answer any questions that may come up, and the audience is welcome to type them in the chat or unmute yourself. And maybe as questions are percolating, I could start off. I think each speaker touched upon it in some capacity, but maybe we could elaborate and speak to the necessity of integrating stakeholders, whether that be the recipients or family members, experiences in formulating research questions or developing clinical care and clinical outcome objectives, policy and economics. So maybe we could expand on that integration and the necessity of it.
Maybe I'll start. I mean, Kathleen really touched on that and brought home to us, yet again, but very importantly, that we need to think beyond just the research question, the hypothesis. These are patients that live every day and every night with their disease states. We're trying to help. We don't know if our interventions, when it's research, are going to help or not. We know that we can cause harm. That's part of research and research experimentation. But I think learning the bigger picture and finding ways to get better communication is really key. And you highlighted that very well for us, Kathleen.
Yeah. And I don't know if, Kathleen, would you like to speak?
I would. It's, I can see both sides of the equation. I have a background in pure science. I have a chemistry degree, and I also have a nutrition degree. And I think it's really important to involve the participants in your research in developing some additional things, because as Dr. Shapiro said, we live with it day in and day out, and our outcomes and goals might be a little bit different than what the clinician is looking for. And if that's excluded, you miss out on an opportunity to have a very rich program.
And I will say that, it's tough sometimes in the basic science to think about how you can integrate patient input. But what we've started doing with our research program over the last year and a half is actually having patient focus groups to help direct us towards gaps that we may not be supporting in the research arena. And one actually that Kathleen touched on, which was brought up by a focus group, was the impact of peri and post-menopause on islet function. And as a result of that, we have actually integrated some of that, some measures, hormonal measures, into the work we're doing. We have a program that's building an atlas of islet biology, and we've actually integrated that into that. And that was a direct result of patient workshops.
Fantastic. I think it's this discussion today has really highlighted the necessity of integrating the patient's experiences in our both clinical and research questions. So again, thank you, Kathleen, for stimulating that perspectives and discussion. And if it's okay, we have time for maybe one more question here. This is from Mary Koch. This may have been mentioned, but what is the minimum cell count that must be present when extracting islet cells from a pancreas for islet transplant? Often pancreases we send to Hope will be declined for islet transplant for insufficient count. So maybe Dr. Shapiro could elaborate on that.
Yeah, I can. So the dosing of islets is not a precise science. You might think it is, but we try to dose, well, for the first transplant more than, and we ideally like more the better, but we can't control it from what we get out from each pancreas, but we like more than 5,000 islet equivalents per kilogram. So that's not cells, that's balls of islets essentially that vary in size from 50 to 500 microns. So the dosing, you're right. So we don't want to initiate a transplant and then underdose the patient. And maybe some of those islets would be just fine. Maybe they'd be more potent. We just don't know that. And maybe we should be transplanting some of the, perhaps that we regard as failures. Maybe those pancreases actually only had that number of islets in them. There's certainly a variable number of islets. Not every pancreas has the textbook 1 to 2 million islets within it. But I'm interested in Pat's comment too.
Yeah, I do have a comment on this, Marcy. I'd like to point out that many of the organs that go to the clinical group and may not then, after isolation of islets, go for transplant, can be used for research. And they do go out for research. And this is one of the wonderful things that the clinical group does. And so, you know, obviously these have to have research consent, but if there's one message I want to get across, and I hope that I was able to in my talk, is that that's not a failure. You know, there is valuable information to be gained from those islets that do not go for transplant.
I'd just like to echo that as well. A lot of the studies that were presented today, so some of those islets came from organs that didn't go to transplant, but had meaningful impact to the research community and addressing clinical care too. Well, I would like to, in the interest of time, thank you for the panel for stimulating some wonderful discussions, and your perspectives are very enlightening. Thank you for staying a little bit later. And also, I would like to announce that this is our last ATI seminar series for the 2021-2022 series. So we will welcome everybody back in September. And again, thank you so much for our speakers today. Really appreciate your time and efforts. Thank you.
Thank you. Thanks, Andrew. Thanks for putting this together.